All right. Chemistry 102, practice exam 5. This is question number 30. This is kind of where the material starts from chapter 22 in our textbook. In chapter 22 is the aerobic respiration and energy production chapter. So question number 30 says, what is the function of coenzyme A? And you know that you should know the structure of coenzyme A, that we have our acetyl group. Here's our acetyl group. Then we have that high energy thioester bond. That's this bond here, which is bound to a sulfur. And then, of course, coenzyme A, right? This is a very big molecule, but this is the shortened version that we like to use. What is the function of coenzyme A? A, to oxidize carbohydrates in glycolysis. That doesn't sound reasonable to me. To carry ADP to the citric acid cycle. No. The reason the first one is wrong, by the way, is because coenzyme A is not involved in glycolysis. So we move over to C. It says to carry acetyl groups in the form of acetyl coenzyme A to the citric acid cycle. And that is exactly correct, right? Because we have acetyl coenzyme A that's going to combine with oxaloacetate in the first step of the citric acid cycle, right? Where we bring the acetyl group into the citric acid cycle. Next one says to carry phosphate groups to glycolysis. No, that's not true. Into hydrolyzed triglycerides, so it's got nothing to do with that. All right, let's move on to question number 31. It says, in which stage of the metabolism is the most energy in the form of ATP produced? Well, it's definitely not in glycolysis, right? We don't make very much ATP in glycolysis, and we looked at how much ATP is produced in glycolysis. It's an ancient pathway. It is anaerobic. It does not use oxygen. Then we have the transcription pathway. No. Citric acid cycle. No. The stage of metabolism where most of the ATP is produced is from oxidative phosphorylation, right? You might be wondering, why isn't it the citric acid cycle? Well, in the citric acid cycle, you form intermediates like NADH, FADH2, but they go through oxidative phosphorylation in order to produce ATP. And so the answer is D. It's not the digestion of food molecules either. All right, let's move on to question number 32. It says, what cyclic metabolic pathway is the final stage for the degradation of carbohydrates, fats, and amino acids released from dietary proteins? Well, this one is one where you just kind of have to know what it is. Well, if it's a cyclic metabolic pathway, it's probably the citric acid cycle, right? This is where we take those intermediates from carbohydrates or fats or amino acids, and we put them into that citric acid cycle, and we extract energy from them in the form of ATP. The next one deals with the F0, F1 complex in oxidative phosphorylation. This is the okay, I'm going to come see it later, but I'm teaching class right now. Okay, I love you. All right. The next one, question 33, is about the F0, F1 complex used in oxidative phosphorylation. And what is the role of F0? So if you read what the F0, F1 complex is for, I mean, I'll just kind of spill the beans here, that the F0 portion, part of that complex, is the channel whereby the protons can return into the matrix from the inner membrane space, right? They're going to pass through the F0 portion, and then that triggers the F1 portion to cause the phosphorylation of ADP to form ATP to occur. So again, if you look at the structure of the F0, F1 complex, and I'm just looking at our book here because Mr. Dion is not the best artist, but if you look at figure, so this is in figure 22.7 from our textbook, you see you have the inner membrane, and then you have the lipid bilayer, and you have the F0 portion, which looks something like this. So here's the F0 portion, and then you have the F1, which again, I'm going to butcher it here, but the F1 portion looks something like this. This would be the F1 portion. Well, the protons end up passing through the F0 portion, and it's the F1 portion that activates ATP synthase and causes ADP to be converted into ATP. All right, let's move on to question number 34. Again, the answer to 33 was C. Question 34, it says, which statement concerning the regulation of the citric acid cycle is true? So we're looking for true statements here. And if you look in your textbook, there's a whole section where it deals with the regulation of the citric acid cycle. So that's section 22.5 in your textbook, 22.5, which is called the control of the citric acid cycle. Well, what's going to stimulate what's going to regulate the citric acid cycle? Well, if we have high levels of ADP, is that going to stimulate the citric acid cycle? The answer is yes, absolutely it will, right? Because we have an energy deficiency, right? We don't have enough ATP. What if we have high levels of NADH? Is that going to inhibit the reactions of the citric acid cycle? Yes, because every NADH has a value of 3 ATPs. And so, yes, that's going to inhibit the citric acid cycle because we have a surplus of energy. Next one, what if you have low levels of ATP? Is that going to stimulate the citric acid cycle? Of course it will, because you have an energy deficiency. D, high levels of ATP inhibit the reactions of the citric acid cycle. Well, these two are opposites. So yes, this is going to be true. And so the answer is, or the correct answer is, that all of these statements, A, B, C, and D, they are all, in fact, true with respect to regulation of the citric acid cycle. Question number 35 deals with the transamination reaction. So this is covered in section 22.7 of our textbook, the section on the degradation of amino acids. And it says, what coenzyme is required for all transamination reactions? And the answer is pyridoxal phosphate is the coenzyme that is used in all transamination reactions. If you go in our textbook and you look at figure 22.10, so you could look at figure 22.10, there you see the mechanism of transamination, and you see how pyridoxal phosphate is required for the transamination process to occur, which of course involves the removal of the amino group from a amino acid. There we go. Question number 36 says the oxidative reactions of metabolism provide for most ATP production. In what specific part of the cell do these reactions occur? I'm going to throw this out there to my students and see if they can answer this question. Where does most ATP production occur? Well, I had a student a number of semesters ago who used to have a picture of a mitochondria on his t-shirt, and it said next to it, the powerhouse, right? It's the mitochondria. If you've ever studied biology before, I know many of you have, they call this the powerhouse of the cell, right? The powerhouse of the cell. And the reason why is because all the enzymes and coenzymes and cytochromes that are responsible for oxidative phosphorylation, they're all in the mitochondria, right? So that's where the majority of ATP is produced, right? The citric acid cycle, all this stuff takes place inside of the mitochondria. That's why the start of chapter 22 begins with a discussion on the mitochondria. Yes, we produce some ATP outside of the mitochondria in the cytoplasm. That's called, and that's the result of glycolysis. However, most of the ATP that's produced is produced inside of the mitochondria. All right, question number 37 says, what is the usual fate of the alpha amino group in the catabolism of amino acids? And of course, that goes right back to section 22.7 in our textbook, which deals with the degradation of amino acids. Well, if you take a look at that section, it discusses the breakdown of a couple of different amino acids, and it shows you what happens to the amino group that is removed from the proteins, or sorry, from the amino acids, which were part of a protein, and they are removed in the urea cycle, which is covered in section 22.8. And so the amino group is converted into urea, which is excreted by the body in the urine. If you look at the structure of urea, so if we look at the structure of urea, it was the first organic compound synthesized from inorganic starting materials. Okay, you can see that you have two amino groups, and one of them would come from the amino group that's removed from an amino acid. All right, question number 38 says, what is the term for the region of the mitochondrion that is enclosed by the inner membrane? I'm going to throw this out there to my students. If anybody could tell me what is the term for the inner region of the mitochondrion, which is the singular of mitochondria. Could anybody tell me which part of the Yes, thanks, Aaron. It's the matrix space, right? It's the matrix space. If we draw, the shape of a mitochondrion, it's like a football shape, okay? So it's a football-shaped organelle, and then you have these kind of folds, and you'll have to bear with my artistry here, but you have these kind of folds here that we call the cristae, like that, okay? So here's my mitochondrion. Here we have the outer membrane. Here, I'll fold it up. This is the inner membrane, like that. Okay. This region out here, outside of the inner membrane, we call this the intermembrane space. So this is the intermembrane space. And then inside here, let me use a different colored highlighter. I'll just point to it. Here we have the matrix space inside. Okay, so that's the matrix space, or sometimes it's just called the matrix. All right, where in the body does the degradation of amino acids occur? This is one of those questions. It's more one of those things where you just kind of have to read it, and just have some of these facts memorized. And the degradation of amino acids actually occurs in the liver. So it doesn't occur in the cytoplasm, fat tissue, or adipose tissue, the stomach or small intestine. It takes place in the liver. And the last question is, what is the terminal electron acceptor in aerobic organisms? Could anybody answer this one here? We have the electron transport chain. The electrons get passed through the lipid bilayer across different enzymes. And then where do they end up after they run out of energy? What is the acceptor of the electrons at the very end of the electron transport chain? Could anybody tell me what's the electron acceptor? Okay, if you look at figure 22.7, it shows you the electron transport chain, and it shows you where the electrons end up, right? As you oxidize NADH and FADH2, you pump the protons across the inner membrane, but then you have the electrons that go across the inner membrane, right? And they cause the protons to be pumped across the membrane space, and then the electrons end up being attached to oxygen. So oxygen is the acceptor, right? The fate of the electrons in the end is that we take half a mole of oxygen plus 2 electrons plus 2 protons, and we end up with H2O. So that's the ultimate fate of the electrons that come from the oxidation of glucose, right? Loses electrons, oxidize. You remember that? Or oxidation is losing from Chemistry 101. So you're losing electrons. When you oxidize the glucose, you're converting the glucose to carbon dioxide. Every carbon is getting oxidized to carbon dioxide, but you're losing electrons. And those electrons are going to combine with protons, right, from the NADH and the FADH2, and they also combine with oxygen to form water. So water is the terminal electron acceptor in aerobic organisms.